In brief, as we follow jet scheme of electron transport, the electrons from water to PS2, that is P680, and then to pheophytin, and through various electron carriers received by P700 molecule of PS1, and from P700 to ferredoxin, and ferredoxin to NADP+. So, as we follow the brief electron transport mechanism here, and this is a kind of jet scheme explained by Hill and Bandol, and no two electrons back to their original home we followed. Electrons of PS2 received by PS1, electrons of PS1 received by NADP+, but the electron loss of PS2 is compensated by water molecule. So, as we tally that electron transport and balance, and here we identified, and finally the pair of electrons received by NADP+ do not cause any loss of electrons in P700 and P680. But finally the loser of electrons that cause loss of electrons to water molecule, because already water molecule split into 2 electrons, 2 protons and half oxygen molecule. Okay. So, here finally who is the loser of electrons? Water molecule. Who is the gainer of electrons? NADP+. So, ultimate donor of electrons is water molecule. Ultimate acceptor of electrons is NADP+. Okay. So, now with this we completed just simply the mechanism of electron transport that occur during non-cyclic electron transport, and that is from PS2 to PS1 and then to NADP+, but the loss of PS2 is compensated by water. So, finally we came to know the electrons of water through various electron carriers including 2 photosystems and the comobile carriers plastoquinone and plastocyanin and cytochrome complex finally added and received by NADP+. So, it is reduced to NADPH. So, here photolysis of water, if one water molecule undergoes splitting and in presence of excited chlorophyll P680 pigment, okay, it causes the liberation of 2 electrons which are involved in non-cyclic electron transport finally received by NADP+ and reduced to NADPH and cause the synthesis of reducing power. Okay. And in addition we observe one more event, that is proton translocation. Okay. Now, let us discuss proton translocation, and here already we followed splitting of one water molecule cause the liberation of 2 protons directly into lumen, and the 2 electrons involved in non-cyclic electron transport cause the shifting of 2 protons from stroma to lumen. Already we followed in detail, and here plastoquinone involvement and cytochrome complex involvement cause the movement of protons from stroma to lumen. As plastoquinone receive electrons as well as protons and pull the protons from stroma, whereas cytochrome complex leave the protons or left the protons, so that protons translocated into lumen. So, finally for a pair of electrons involved in non-cyclic electron transport, we observe 4 protons added to lumen. So, gradually as we observe the event, subsequently large number of protons may be added to lumen, and that cause high concentration of protons in the lumen, whereas reduction in proton concentration in the stroma, because time to time protons translocate from stroma to lumen. A proton concentration gradients developed according to Peter Mischel chemiosmotic theory, as we follow that proton concentration gradients involved in the synthesis of ATP, as here 4 protons which are accumulated into lumen during non-cyclic electron transport of a pair of electrons, that is by splitting of one water molecule, they translocate and they are channeled through coupling factor, and here this is CF0 and CF1 part of coupling factor, and the protons channeled through CF0 and finally crossed the head region, that is CF1. Here there is an enzyme called ATP synthase is located, and here due to the translocation of protons or channeling of protons through the head region, certain amount of energy get liberated here. That energy is utilized to couple ADP and pi to form ATP, particularly the energy of two protons. The energy of two protons utilized for the coupling of one ADP and pi to form one ATP. But here, as a result of non-cyclic electron transport of a pair of electrons, 4 protons are channeled through coupling factor, and that 4 protons again added to stroma. But meanwhile, the energy liberated may utilized for the synthesis of 2 ATP as explained by Peter Mischel. So here electron transport also caused the synthesis of ATP as it caused proton translocation also, that is due to the involvement of plastoquinone and cytochrome complex. Okay. So now let us follow what are the energetics of that pair of electrons involved in non-cyclic electron transport. So students, now let us follow the energetics that are formed during non-cyclic electron transport of a pair of electrons, and here is the equation that explains, and here we followed as one water molecule undergo photolysis and due to that excited P680 pigment and liberate a pair of electrons. That pair of electrons involved in non-cyclic electron transport, and of course both photosystem 1 and photosystem 2 get excited here. So for the non-cyclic electron transport of 2 electrons, 4 photons are utilized here, and that cause the synthesis of 1 NADPH, that is the reducing power, as well as, and here that pair of electrons involved in NCET, and they cause addition of 2 protons directly to lumen as well as 2 protons through plastoquinone and cytochrome complex, that is from stroma to lumen. So totally 4 protons added to lumen. If that 4 protons channeled through coupling factor and finally added to stroma, and meanwhile that energy liberated caused the synthesis of 2 ATP molecules, and due to photolysis of water, not only 2 electrons, 2 protons and half oxygen molecule as byproduct evolved. So if one water molecule involved in photolysis in light phase reaction and cause a pair of electrons, that is 2 electrons in non-cyclic electron transport by utilizing 4 photons, and what are the energetics that are produced is 1 NADPH molecule, 2 ATP molecules and half oxygen molecule as byproduct, and these are the energetics of non-cyclic electron transport of a pair of electrons as a result of photolysis of one water molecule. Students, now we are in the discussion of light phase of photosynthesis, and there the main event electron transport mechanism, and there are 2 types of electron transport I mentioned, non-cyclic electron transport and cyclic electron transport. Already we completed non-cyclic electron transport and its energetics. Now let us focus on cyclic electron transport and what are the subsequent results. Now let us see cyclic electron transport mechanism, and here in cyclic electron transport only PS1 is involved. So the reaction center pigment, that is chlorophyll A of P700 type, that absorb longer wavelength, and as it absorb 2 photons get excited and liberate 2 electrons. That 2 electrons liberated from P700 molecule from the reaction center of PS1, and they are received by an unknown compound located in light harvesting complex of PS1, and then to ferridoxin, the iron sulfur protein and electron carrier receive that pair of electrons, but from ferridoxin the electrons are not received by that FNR, mean that NADP reductase enzyme, because it is not available in this area. So the electrons from ferridoxin back to cytochrome complex, and they are received by cytochrome B6 of cytochrome complex, and from that cytochrome B6 the electrons are pulled by plastoquinone. As already we said, plastoquinone is the mobile electron carrier, and that lie between that cytochrome complex and PS2, and here from cytochrome complex that plastoquinone pull that electrons, and in addition to electrons To reduce into plastoquinol, it require protons also, and it take electrons from cytochrome B6 and protons from stroma as usual. So here plastoquinone, the mobile electron carrier, that take electrons from cytochrome B6 and protons from stroma, and again that proton uptake by plastoquinone is an active process we consider. And now plastoquinone reduced to plastoquinol and immediately get oxidized and liberate that electrons and protons, and the electrons are received by cytochrome F. But of course cytochrome complex is only electron carrier but do not receive protons. So the protons are left by cytochrome F, it do not receive that protons, and that protons now added to lumen and cause translocation of protons from stroma to lumen, and by the involvement of plastoquinone and cytochrome complex here. For that 2 electrons, 2 protons added to lumen, and then the electrons from cytochrome F, and they are received by another mobile electron carrier nearer to P700, that is nearer to PS1, plastocyanin, a copper containing electron carrier. Now give that electrons to excited P700. Now that excited P700 molecule at ground state, so that electron transport mechanism is completed. So here as we conclude, the 2 electrons liberated from P700 molecule through various electron carriers including plastoquinone, and finally back to their original home, and finally back to that excited P700 molecule. So it is a kind of cyclic transport of electrons, and that is cyclic electron transport. But during this cyclic electron transport of that pair of electrons, a pair of protons, 2 protons translocated from stroma to lumen. Okay? And here as electrons back to their original home, that is P700 molecule, and there is no chance for the reduction of NADP plus. Moreover, there is no enzyme available in this area. And here the 2 protons which are channeled from stroma to lumen, and later they pass through coupling factor and again added to stroma according to Peter Mischel chemiosmotic theory. The energy liberated by the translocation of 2 such protons utilized for the synthesis of 1 ATP. So we may estimate the energetics of cyclic electron transport as a pair of electrons involved in cyclic electron transport and cause the translocation of 2 protons, and which are channeled through coupling factor and cause the synthesis of 1 ATP. And this is the equation that represents the energetics formed during cyclic electron transport. Pair of electrons back to their original place, it is cyclic movement of electrons, but at the time and 2 protons from stroma translocated to lumen may help in the synthesis of 1 ATP molecule. And now let us compare, and here what happened during cyclic electron transport when compared to non-cyclic electron transport. Here the participation of only PS1 we observed. So there is no participation of PS2. PS2 is not involved in cyclic electron transport, and there is no photolysis of water and no oxygen evolving complex involvement. So oxygen evolving complex as well as photolysis of water does not occur, and so there is no evolution of oxygen, okay, and as well as there is no NADPH formation because the electrons are back to P700 molecule and involved in cyclic electron transport. And the energetics we expect only 1 ATP molecule as 2 protons translocated from stroma to lumen and later channeled through coupling factor. That is about cyclic electron transport. Now let us follow the differences between non-cyclic and cyclic electron transport in the form of a table, and later we conclude the energetics. Students, as already we followed the mechanism of non-cyclic electron transport and cyclic electron transport in detail, let us take a glance how they differ with one another. And here we listed out the differences between non-cyclic electron transport and cyclic electron transport. In non-cyclic electron transport, there is the involvement of both PS2 and PS1, and here in cyclic electron transport only PS1 involvement. And in non-cyclic electron transport, OEC, which is always associated with PS2, and photolysis of water, that is involvement of oxygen, splitting of water molecule is there, and such events are not happened during cyclic electron transport due to the non-involvement of PS2 and OEC, and oxygen is evolved as byproduct during NCET, but there is no oxygen evolution in cyclic electron transport because there is no photolysis of water. And during non-cyclic electron transport, finally the electrons are received by NADP plus, so reduced to NADPH. For one water molecule that undergo photolysis, one NADPH molecule is reduced, mean formed, and that is the reducing power synthesized. But during CET, no involvement of water, no OEC, and moreover electrons back to PS1, that is P700 pigment, and so here there is no NADPH formation. Okay, but as we compare the energetics, ATP is the common energetic formed during both non-cyclic and cyclic electron transport mechanism, but here for a pair of electrons involved in NCET, as both the photosystems are involved in excitation and mean excitement, so 4 photons, that is the light energy, 4 photons utilized and cause the translocation of 4 protons, 2 protons from water and 2 protons from stroma, as they are translocated through coupling factor cause the synthesis of 2 ATP molecules. But whereas in CET, for a pair of electrons involved in cyclic electron transport from P700 again back to P700, and only 2 photons utilized for the excitation of PS1 and only 2 protons translocated from stroma to lumen through Q cycle, and that caused the synthesis of only 1 ATP. And these are the differences between non-cyclic electron transport and cyclic electron transport, which help you a lot to follow the objective orientation questions. Okay? And with this we concluded that electron transport mechanism is the major event in light phase reactions and which cause the involvement of the synthesis of assimilatory power. But as we conclude, how that electron transport cause the synthesis of ATP. Here, this is the area that we have to focus further, and actually we followed electron transport mechanism majorly here, but how that electron transport cause the synthesis of ATP, that is by proton translocation, explained by a scientist called Peter Mischel. So now let us follow Peter Mischel's chemiosmotic hypothesis, which explains photophosphorylation, that is light induced ATP synthesis and favored by electron transport. So now, and the topic is photophosphorylation. Okay? So students, now our focus is on photophosphorylation, how light induces synthesis of ATP, because already we followed light induces electron transport mechanism, and during non-cyclic electron transport, electrons added to stroma that cause reduction of NADP plus to NADPH. So NADPH formation is clearly explained by electron transport mechanism, particularly in non-cyclic. But as we follow how that electron transport cause proton translocation, how the proton translocation it promote ATP synthesis, and we need further explanation for that, and that means to follow that photophosphorylation, light induced ATP synthesis. As we analyze this word, phosphorylation means synthesis of ATP by coupling ADP and inorganic phosphate, and photo mean light. So light induced ATP synthesis, photophosphorylation, how it occur? To follow that we need a hypothesis, and that is chemiosmotic hypothesis proposed by Peter Mischel. According to Peter Mischel's hypothesis, that is chemiosmotic hypothesis, chemical osmosis of protons, that is proton concentration gradients is the main force or driving force for the synthesis of ATP, he explained. Now the explanation of Peter Mischel, that is chemiosmotic hypothesis, and how it is applied to non-cyclic and cyclic photophosphorylation, here we follow. And in non-cyclic electron transport mechanism, we observe the involvement of PS2 as well as PS1, and of course that cytochrome complex including plastoquinone. and as a pair of electrons from water molecule involved in non-cyclic electron transport finally added to stroma and utilized for reduction of NADPH. Meanwhile, directly 2 protons from water molecule added to lumen by photolysis of water and 2 more protons, and they are channeled, mean translocated from stroma into lumen by the involvement of plastoquinone and cytochrome complex, as plastoquinone actively uptake that protons, but cytochrome complex do not take or receive the protons and leave the protons into lumen, so that for a pair of electrons involved in non-cyclic electron transport, 4 protons channeled, mean added to lumen. So lumen here now act like a proton reservoir, and gradually as the process happens and there is reduction in the concentration of protons in stroma and increase in the concentration of protons in lumen, and that cause proton concentration gradients between lumen and stroma across the thylakoid membrane. Okay, so there is a proton concentration gradients, high concentration of protons in lumen, low concentration of protons in stroma due to this proton translocation as a result of electron transport observed here, and that is called proton concentration gradients by Peter Michell, and this is the driving force. How it drives, let us see. As proton concentration gradients develop between lumen and stroma, and so now due to proton motive force, now the protons from lumen try to move down into stroma, but the thylakoid membrane is impermeable to protons. So the protons are always channeled through coupling factor, that is ATP synthase complex, which has 2 parts, that is CF0 and CF1. And now that accumulated protons in the lumen, okay, they are channeled through coupling factor as thylakoid membrane is impermeable. So the 4 protons channeled through coupling factor and they cross the head region and again added to stroma, but meanwhile at the head region, and that is called catalytic site, with the enzyme ATP synthase, this jumping of protons, they cause liberation of certain amount of energy, and that energy couple ADP and inorganic phosphate molecules to form ATP. As here non-cyclic photophosphorylation we are following, we represented for a pair of electrons involved in non-cyclic electron transport, and 4 protons, they are translocated and channeled through coupling factor and cause the synthesis of 2 ATP molecules. Among the 4 protons, 2 protons directly by photolysis of water added to lumen, and 2 protons through non-cyclic electron transport of electrons, that is by the involvement of plastoquinone and cytochrome complex. So totally for a pair of electrons involved in NCET, 4 protons added to lumen and they are channeled through coupling factor, again added to stroma, but meanwhile their energy is captured and utilized for the synthesis of 2 ATP we followed. And whereas cyclic photophosphorylation as we see, and here only PS1 involvement, this is PS1 and PS1 involvement and cause that electron transport in cyclic manner, but during that cyclic electron transport due to involvement of plastoquinone and cytochrome complex, again here a pair of protons from stroma added to lumen, and gradually as the process may occur repeatedly and there is proton concentration gradients between lumen and stroma developed, and the protons now try to move down from lumen to stroma due to proton motive force, but as thylakoid membrane is impermeable, and so that and the protons always channeled through coupling factor and finally added to stroma, but meanwhile the energy liberated by that protons utilized to couple ADP and pi to form ATP. So here as we follow photophosphorylation process as explained by Peter Michell based on chemiosmotic hypothesis, and that is proton concentration gradients developed between lumen and stroma is the main driving force for ATP synthesis, and the property of thylakoid membrane, that is impermeable to protons. So, protons always channeled through coupling factor, and that is another reason and why that energy is utilized for the synthesis of ATP. So, for a pair of electrons in NCET, the energetics to ATP, for a pair of electrons in cyclic, and cause the synthesis of only one ATP. And that is about Peter Mischel's chemiosmotic hypothesis, how it caused phosphorylation, that is ATP synthesis. So, light induced electron transport cause proton translocation. Proton translocation that leads to proton concentration gradients, but as thylakoid membrane is impermeable, the protons always channeled through coupling factor, and as the protons cross the head region of coupling factor with an enzyme, that is ATP synthase, and that energy liberated by the crossing protons or channeling protons captured by ATP synthase and utilized to couple ADP and pi to form ATP. So, light induced electron transport cause or help in synthesis of ATP as explained by Peter Mischel chemiosmotic hypothesis. Okay. So, students, with this we completed that major part of light phase mechanism, and that is the important event electron transport and two types of electron transports, how electron transport cause proton translocation, how that proton translocation based on Peter Mischel's theory help in ATP synthesis, that is photophosphorylation. So, the aim or the significance of light phase reactions is synthesis of NADPH and ATP combinedly can be referred as assimilatory power. So, synthesis of assimilatory power is completed during light phase reactions, and what happened to that assimilatory power, where it is consumed, and we follow in the next session. And meanwhile, and we have to focus on the difference between bacterial photosynthesis and higher plant photosynthesis in detail, because and in this particular concept and many times we are getting questions in the papers. So, now and we continue the discussion on differences between bacterial photosynthesis and higher plant photosynthesis. Then in the next session we follow that dark phase reactions where the assimilatory power is utilized. So, now let us take a glance on differences between bacterial photosynthesis and higher plant photosynthesis.